We're going to be doing some work on the ISAs, the Investigative Skills assignments. That's going to be coming up for us soon. And I thought it would be useful to show you how these things are marked, how the mark schemes work, how the marks are awarded. Now, in this setup, we've got the student's work on the left here, and we've got the mark scheme on the right. And got here award 1 mark for all shaded squares correct. This basically is the setup of the experiment, a very simple one at the start, and for getting that information correct, we'll check across there. They've got the same numbers, and then 16, 12, 8, 4, 16, 12, 8, 4, 0. The student has got all of that information correct. There's 1 mark, so 1 mark is awarded. Now in this next section, what the students have done is they've recorded their data in a table in the space below. What they've had to do is they've had to decide which particular variable goes here and which variable goes here. Also, how to present the information. You'll notice there are some negatives here. That's very important. Now here's the marking criteria. Students' data presented clearly with distance plus or minus sign and concentration of sucrose clearly indicated. Well, is that there? Yes. Well, there are minus signs. That's supposed to be a plus, and so is this one. And the concentration of sucrose in moles per decimeter cubed, and distance between ends of strips, and I just believe that's centimeters or millimeters, so I can give a mark for that. Concentration is in the first column. Now the concentration is the independent variable. That's the one that you've controlled, and the convention is that that will be the first one that is in the table of results, and it's given here. Concentration is going to be in the first column, but if the table is horizontal, concentration should be in the first row. Well, this one is horizontal. There's the first row and there's concentration, so I can give a mark for this. For the third mark, units clearly stated and given only in column headings. Well, the units are clearly stated here and they're only in the column headings or on the row endings. If they'd put, for example, centimeters, centimeters, centimeters in these boxes, then they would not have got that point. The convention is that the units are given clearly at the start in the column headings, but not in the individual data entries. Okay, now we're going to look at how the graph of the results is marked. And firstly, the title incorporates both the dependent and independent variables. So what we're looking for here is distance or length between the ends and the concentration of sucrose. So what we've got here is a graph to show how different concentrations of sucrose solution can affect cells from a plant stalk. Now, is there anything in there about distance? No, there isn't. So one of these variables, the dependent variable, is missing. So this gets no marks. The next mark, the distance on the y-axis and the concentration on the x-axis. This again, this is a convention. The distance will be the dependent variable, the concentration will be the independent variable. The independent variable goes on the x-axis. Well, let's see if it managed to do that. So we're looking for distance on the y-axis, yes, and that is there, and concentration on the x-axis, and there it is. So they have a mark for that. The next one, both axes are labelled to indicate units, and there we have millimeters, and there we have percentage of sucrose solution. So there's a mark noted for that. The next one, appropriate scales selected for both x and y axes. Now, appropriate scales as a general rule means that you're using between 50 and 75% of the available space. You certainly shouldn't be using less than 50% in any of the axes lengths. So let's have a look. Well, this one is about, well, 3/4 of the x-axis, and it's about 50% of the y-axis. So I'm going to award a mark for that and say that appropriate scales have been selected for both x and y axes. Okay, so now I want to check the accuracy of these graph points. And to do that, I'll need to go back to stage 1, get the actual data that they've used to make the graph, do a little screen capture, put that to one side. Okay, let's bring this in and see how good the graph is. Okay, so for a value of 0, we've got 70 millimeters. For 0.2, we've got 81. For 0.4, we've got 70. For 0.6, 0.67, 0.8, 64, and for 1, 62. Right, I'm quite happy with that. Let's see how many marks we can award. I'm happy that all points have been plotted accurately, so I can award 1 mark for that. I'll type in here accurate plotting and award 1 mark. Now to the data being presented as a line graph, and it says depending on the data obtained by the student, points should be joined by the best fitting curve if it's considered that intermediate values are likely to fall on such a curve. And we look at this section, we look at this, we look at this part of the curve, the part of the data, it does look like a curve. So I'm quite happy to give that a mark, and that will be correct presentation. Give that a 1, and so that's 5 out of 6, and the mark that was lost was not fully labeling the graph. So 5 marks, confirm. Okay, so we're on to section A, and again we're looking at the student work and the mark scheme. Now, the question: you were told to put lids on the petri dishes before leaving them. Explain why it was necessary to put the lids on the petri dishes. Now, this student has answered so that no extra solution was added by mistake, which would affect the concentration and results. Well, you could get very much the same effect by putting a sign in front of the petri dishes saying do not add any more solution. The petri dish lid is to prevent evaporation of water, and that will keep the concentration of sucrose the same. They haven't got anything like that, so it's no marks for this part. So I don't need an annotation, I'll just put a 0 in there. And we're on to our next question. The cells of different species of plant contain different concentrations of sugars. Now remember that bit because they've actually given you that information. Use this information to explain why you may get different results if you used another species of plant. And then what he does is completely restates what has just been told to him, and you get no marks at all for that. If we used another species of plant, we might get different results because the different concentrations of sucrose solution would affect the plant differently, as the plant would contain different concentrations of sugar. There's no explanation there at all. What he's just basically done is said the same things that are already in the question, so no marks for that. What they were looking for is that the different concentrations of sugars would affect the water potential, and the water potential would affect the movement of water into or out of the cells. And here in the comments, because the new sugar concentration is not specified, it could be movement into or out of, and therefore giving an increase or a decrease of the amount of water in the cells. This is an actual explanation. This isn't. This is just saying the same things in the question back again and hoping that somebody gives them a mark for it. So let's give this a mark, and it's going to get nothing. Now, question 3. You were told to put thin strips from the stalk into the solutions. Would the stalk curve if you put uncut stalks into the solutions? Explain your answer. Well, it's an old standard question in biology, this one. And what you've done here, this is a stem that's been cut into longitudinal strips. And the point about this is the thickened section, the black section there, that's the outside layer of the stem, and that's waterproof. Water cannot penetrate it. So if this black line, this thick line, is the outside, and these are the inside cells, and these ones are permeable to water, they will change their volume when they're put into hypertonic or hypotonic solutions. Now this one was put into distilled water, and with distilled water you would expect water to enter the cells and make them swell, and they swelled up, but the cells stuck on this outer layer, they were unable to absorb any water. They stayed their original size, so you get this curvature. This piece of stem was put into a concentrated sugar solution. You would expect the water to leave the cells, the permeable cells here, and shrink. These cells here near to the outer layer, they're not permeable, and so you would expect them to stay the same size. If you can imagine that this was a brick wall, and all the bricks on this side of the wall expanded, they all got bigger, then you would see a curve in the wall. Okay, so that's explained how you would expect to see a curvature with thin strips from the stalk. Now the question is actually asking us, would the stalk curve if you put uncut stalks into the solutions? In other words, instead of cutting the stalk into these strips, you put the whole thing into the water. Would you expect to see a curvature? And the answer is no, you wouldn't, because all the cells would change length by the same amount. Has he got that? He's put no, as water cannot enter the cell as the cell walls do not allow it. Well, the mark is only for the explanation. He's got the answer right, but is the explanation right? Unfortunately, no marks for this. He's put, as water cannot enter the cell as the cell walls do not allow it. And what he should be saying is because this outside layer is waterproof. So, right answer, wrong explanation, no marks. And I'll annotate this here. Wrong explanation.